Okay, good morning and happy Wednesday. Today is March the 25th. This is the second of the two videos that I'm going to be making for today's lesson on concept 14.3. Inheritance patterns are very often more complex than even Mendel would have predicted based on his statistical analyses. Now, um, one example in particular of this kind of thing is multiple alleles. And although Mendel, in his work, essentially said that, or based his work on traits that only had two different alleles, purple and white, or round or wrinkled, or yellow and green in terms of the pea plants, most genes really come in multiple allelic forms, more than two. A great example of this is blood types in human beings. There are four phenotypes, all right? I don't know if you know your blood type. If you gave blood at the blood drive last month or the month before, you may now know what your blood type is. But blood types come in human beings in four major categories. You've got blood type A, blood type B, blood type AB, and blood type O. Um, and these blood types are determined by combinations of three different alleles. Now, remember that each person can only carry two of those alleles, but there are three alleles out there. Those alleles are the A allele and the B allele, both of which are dominant, and the recessive allele, which we refer to as lowercase I. Notice the symbols for them. They're symbolized with a capital I with an A over it. That's blood type A allele, blood type B allele, and then the recessive allele here. So, blood type A is a phenotype that you can get in two ways, either two dominant A alleles or a dominant A and a recessive I. Blood type B is or can be seen in people who are homozygous dominant for B or heterozygous for B and the recessive allele. I don't know why that does that. Blood type AB is when you have one of each of the two dominant alleles, and blood type O is when you are homozygous recessive. And so that's the mix there. And so blood types are determined by which alleles you get from each of your parents. Another this is fascinating genetic effect is that most genes actually don't just have effects on the one thing that they're coding for, but they can have other effects as well. And this is a property called pleiotropy. Now, in pleiotropy, the allele that codes for something oftentimes will have other effects, maybe even unseen effects. The Siena students that I teach examine pleiotropy in fruit flies, and fruit flies who are born or have a genetic phenotype to have no wings oftentimes are very sickly. Right? It has nothing to do with their wing type. They just don't survive as well as a wild type fruit fly. And so whatever the gene is that codes for those wingless fruit flies, that gene has other ill effects on their health. Examples of this in human beings include diseases like cystic fibrosis and sickle cell anemia, which we'll talk about in more detail a little bit later on. Now, some traits may also be determined by two or even more genes. There's a term called epistasis, where one gene affects the phenotype of another gene due to interaction of their gene products. And then polygenic inheritance, where multiple genes affect a single trait. So let's talk about epistasis first. A great example of this is found in Labrador Retrievers, dogs, and other mammals as well. But epistasis is when the expression of a gene at one point affects the phenotype of a gene at another spot on the or in the genome. In labs, coat color depends on two different genes. Um, so you can have a black lab, a brown lab, or a yellow lab. All right, and they're all Labrador Retrievers, but their coat color depends on two different genes. One of the genes is codes for the color of the coat, and those genes are either the black allele or the brown allele, black being dominant and the brown allele being the recessive allele. The other one is a gene that codes for whether or not pigment ends up in the hair. And so if you've got a dominant allele, then your fur will be colored. If you don't have a dominant allele, in other words, two recessives, then your hair won't have any color. And that's how you get a yellow lab. Let me show you. So here we show the mating between two heterozygous black labs, and they're heterozygous for both of these genes. So their genotype is big B, big E, little e, and if they're mated, we would expect the 9:3 to 3:1 ratio that you would find in Mendel's F2 dihybrid cross. However, that's not the case. In this case, you end up producing sperm and eggs in these ratios. 1/4 will be two dominant, 1/4 will be little b big E, 1/4 will be big B little e, and 1/4 will be both recessive genes. And when you put them together, you see, well, this dog should be black, and because it's got a dominant allele, it is black. This dog should be black. It's got the dominant E allele, so it's black. And so same for these two. All right, even though in this case and this case, you've only got one copy of the dominant E allele. Same thing is true for this one. But here, now I've got two recessive alleles for brown coat, but the pigment in the hair is there, so it's a brown lab. Another black lab, another brown lab, black, black, and then here, this is where the other interesting thing ends up. Here, I have what should be a black lab because it's got two dominant black alleles. But in the hair pigment gene, we've got two recessive alleles, and so we end up with a yellow lab. Same thing for this guy and these two. This one should be a brown lab, but it doesn't have a dominant E allele, a hair pigment allele. And then you can figure out the others there. But we end up with a ratio of 9:3:4 in this case. 9 black labs to 3 brown labs to 4 yellow labs. And so if you look at this carefully, you'll realize that brown labs can reproduce with each other and produce brown labs, but they can also produce yellow labs. Yellow labs can only produce yellow labs, and black labs can produce pretty much anything. So it's an interesting thing. Epistasis is this phenomenon. Another aspect of this is polygenic inheritance, and these are traits that vary in a population in a continuum, and this is typically known as polygenic inheritance, which is the additive effect of 2 or more genes on a single phenotype. Human height is a great example of polygenic inheritance. There's over 180 identified genes that affect height. But skin color in human beings, is also something that's controlled by many separately inherited genes, 6 that we've identified thus far. And so there are, for 2 heterozygous organisms, there are 8 possible gametes to be produced. And you can see the combinations of gametes represented here as all light colors and here as all dark colors. And so since each parent is donating 3 of these alleles, if you have 2 parents that are very fair, you're going to end up with very fair kids. If you have 2 parents that are very dark-skinned, you're going to end up with very dark-skinned kids. And any combination in between. Um, the last thing that I want to talk about here is when you end up with an environmental impact on the phenotype. The two flowers that are pictured in this slide are exactly the same species and could have come from the same parental plants. However, because one is grown in basic soil and the other is grown in acidic soil, they have very different phenotypes. And then if you throw in polygenic inheritance, multiple genes for flower color, you can get a wide range of colors in these flowers. All right. This is what's known as a multifactorial inheritance pattern. Okay. So to conclude, an organism's phenotype includes its physical appearance, its internal anatomy, physiology, behavior, and a variety of other things. But the phenotype reflects its overall genotype, not just one gene per se, and its unique environmental history, right, where it's growing up, so to speak. All right, so that concludes my discussion of section 14.3. Enjoy.